Study we're presenting is in Oscillation-Spindle coupling and memory formation. Oscillation-Spindle coupling is a synchronization of two specific patterns of brain activity, slow oscillation and spindle oscillation, which is crucial for successful memory consolidation. Recent findings have indicated that the precise coordination of Oscillation-Spindle coupling deteriorating over the lifespan, contributing to age-related memory decline. And we're wondering if the similar role applies to brain maturation and how Oscillation-Spindle coupling is initiated, since growing and aging go through similar transition in sleep structure and cognitive ability. Additionally, because the majority of early studies in the field concentrated on adults, Han and his colleague decided to conduct a study to look into the connection between Oscillation-Spindle coupling and memory formation in kids and teenagers in order to fill this knowledge gap.
The researchers chose a longitudinal study design in which they tested the memory performance and recorded sleeping brain activity of 33 youth at 2 separate times. Memory performance was measured using an established word pair task. The task had participants encode matching sets of unrelated words, and recall performance was assessed after a short delay and again after sleep intervention. Additionally, the researchers tested the memory performance of a control group after a wake period as a separate measure on the impact of sleep on memory. Sleeping brain activity was recorded using EEG, and data was processed by experts using a brain vision analyzer too. EEG is cost-effective, easy to implement, and temporally fast. However, the data it reveals is not always the most refined. To compensate, EEG data was 1F fractal disentangled and filtered to detect frequencies and peak signature to slow oscillations and spindles. With slow oscillations and spindles detected, the researchers focused on recording slow oscillation spindle co-occurrence as classified by slow oscillation events coinciding with spindle peak frequencies.
The first finding was that the coordinated activity of large neuronal populations, or slow oscillations, and bursts of neural oscillatory activity regulated by the thalamus during sleep, or spindles, became more connected as an individual aged. This indicated improved recall performance and sleep-dependent consolidation in declarative memory. The second finding was that there is a precise temporal slow oscillation spindle coordination that hints at the development of the hippocampal neocortical memory systems. Third was that slow oscillation amplitude and frequency decreased from childhood to adolescence. The fourth finding was that coupling strength, which is the strength of the linkage between slow oscillation and spindles, increased from childhood to adolescence in almost all of the electrodes measured. This increase showed enhanced sleep-dependent memory consolidation, which is related to overall improved recall performance. The fifth finding was that behavioral memory metrics, which are tests that examine your memory function in relation to your daily life, were better at predicting the strength of slow oscillation spindle connections than the amount of spindles present.
In conclusion, during brain maturation from childhood to adolescence, coupling of slow oscillation and spindles becomes more precise, allowing more fluent communication in the memory networks. This more precise coupling indicated better recall performance and sleep-dependent consolidation in declarative memory tasks. And the results of the study that precise spindle coupling constitutes a key mechanism to memory formation. Moving forward, there's evidence of ways to entrain SO and spindles to improve memory performance. If we're able to entrain the SO spindle synchrony, then we may be able to help alleviate memory deficits. It would also be relevant to investigate how tau pathology in the medial temporal lobe, such as in Alzheimer's, impairs spindle SO coupling. This could help decipher a pathway for age and disease-related cognitive decline that could be used to form intervention strategies to improve the related memory deficits.
One limitation of the study was that it was a small study sample. It was only done on 33 people, which does prevent generalizability in the larger population. Out of 33 participants, 23 were female and only 10 were male. This baseline characteristic imbalance can affect pre-specified outcomes. For the second limitation was, while they accounted for variables like oscillatory signatures of NREM2 and 3 sleep and baseline cognitive abilities were assessed by the Wechsler Intelligence Scale for children and adults, there are still confounding variables unaccounted for that can influence memory consolidation, such as mental disorders. For the strengths, one of the conclusions reached was that during adolescence, memory consolidation is superior after a sleep retention interval compared to a wake retention interval, indicating a beneficial effect of sleep on memory consolidation. In a separate condition, the conclusion reached was that from childhood to adolescence, slow oscillation and spindles become more precisely coupled, and this phenomenon improved recall performance and sleep-dependent consolidation in a declarative memory task. Overall, these specific conclusions are supported by the research. The study connects to the course quite well, as there's been a great deal of information taught in lecture and textbook about how vital the hippocampus is for memory consolidation, and this study focuses on the specific brain area for study of the role of sleep spindles in memory. The information that we have learned in this course thus far was quite helpful and supplemental for reading this research piece, as our learned information in class with respect to the crucial role of the hippocampus in declarative memory assisted our understanding and intrigue for this study.